Charles Chiu. I think really excited to have Charles here because one, he's a in fact exemplary translational research scientist, right? So he's an MD, Ph.D, doing really innovative things, particularly on the diagnostic side. So we're talking. This is a conference about Lyme disease and the area of precision medicine. Well, it's really hard to do precision medicine if we don't have the next generation diagnostics that help us measure these patients, characterize these patients, and give us the features that we need to understand and model, personalized individualized treatments for patients. So with that, I'm going to give the stage to Charles, and welcome everybody.
I'd like to thank Joel and the organizers for inviting me to speak with you today. And I'll be going over a lot of material, and feel free to have some questions at the end. I'm going to be talking about metagenomic sequencing in a very broad sense, and how we've been able to take this new technology and be able to use it to develop novel diagnostics for Lyme disease and tick-borne infections. So this slide goes over, it's based on a review and published in BMC Medical Genomics. It goes over kind of the general principles of personalized medicine. Personalized medicine or precision medicine has transformed many different fields, including, for instance, cancer diagnostics for identifying specific gene mutations, or prenatal testing, for instance, for identifying genetic mutations. And it's our hope that, or it's my hope at least, that we can develop personalized medicine for use in infectious diseases. And the general idea is that we want to leverage the omics technologies that have been developed and honed over the past couple of years, including genomics, transcriptomics, proteomics, epigenomics, metagenomics, metabolomics, etc., be able to analyze them in a framework, in a bioinformatics framework, be able to generate complex disease networks and disease patterns that we can then use directly for clinical care. And it's based on the principle that every patient is unique. Patients have different responses to the same infection, different responses to the same drug, different responses to the same disease. And this is why it's really important that we not only take into account just detection of, say, an organism, but also we take into account the patient. So I'm going to very briefly go over a summary of Lyme disease. You'll probably get a slide similar to this in every presentation, but Lyme, I want to emphasize really that Lyme disease is the most common vector-borne disease in the United States. There are probably over 300,000 estimated cases in the US annually, and reported cases is only on the order of 30,000. So typically there's a gross underestimate with respect to the number of cases of Lyme disease. There are over 3.8 million diagnostic tests for Lyme disease performed in the US, at least as of 2008. Nearly all of them are based on two-tiered serological antibody testing. It's caused by the spirochete Borrelia burgdorferi, through the bite of infectious tick of the Ixodes species. The typical acute presentation marked by a bullseye rash, also known as erythema migrans. Late-stage disease can present with a number of complications related to Lyme disease, listed below. And I want to emphasize that there are typically 3 stages of the disease that have been described. So there's an early acute stage, an early disseminated stage, and a late disseminated stage. I'm going to be focusing, for the purposes of this talk, on diagnosis in the early acute stage. So these are patients, for instance, who may have had recent tick exposure. They present with fever. They may or may not have an EM rash. How do we diagnose the disease in these patients? I mean, they could potentially have Lyme disease, especially if they're from an endemic area, but they potentially could have a wide variety of other infectious or non-infectious disease syndromes. This is a summary of kind of two-tiered serological testing. Typically, the first test is an enzyme immunoassay or an immunofluorescence assay. Depending on if the result's positive or equivocal, it then proceeds to a second confirmatory test, which is typically a Western blot test. I'm not going to go over more than that, but I do want to highlight, though, some of the key diagnostic challenges of Lyme disease. So one is that the rash is only seen in perhaps roughly 2/3 of patients. So on the basis of fever and a rash, which is typically the clinical, the way we diagnose Lyme disease in the clinical setting, is clearly inadequate. Antibody production, serological detection using gold standard testing can actually take up to 3 to 6 weeks to become positive. So it complicates the diagnosis of acute Lyme. Even direct detection methods such as PCR or direct antigen testing have reported about 20 to 60% sensitivity in in acute phage, so it's not a good test, unfortunately. Now, I want to highlight actually that PCR testing is considered to be the gold standard for acute infections, for many acute infections, including other tick-borne diseases such as ehrlichiosis, anaplasmosis, or babesiosis, for instance. But for Borrelia, or for infection from Borrelia, it does not appear to be a good test, at least from blood. The sensitivity of serology, which is really the gold standard, or the current gold standard, ranges from about 30 to 70%, depending on the paper that you look at. I can list there some of the problems, including the fact that many of the results can be have subjective interpretation. And really importantly, the tests don't detect other tick-borne infections. So these are tests for Lyme disease, and they're not good tests for Lyme disease. So clearly we have some key diagnostic challenges. Another problem with not having a good test for Lyme disease is the fact that we do not have good diagnostic tests to monitor, for instance, patients' responses to treatments in clinical trials. We don't have a good biomarker for Lyme disease to be able to follow patients who have both acute and chronic manifestations of Lyme disease. And really, it's diagnostics which is, I believe, key with respect to deciding how best to manage or treat these patients. So it's our goal, actually, here initially, we wanted to apply an omics technology called transcriptomics. And the general idea of transcriptomics is we're interested in looking at the global gene expression profile of patients who are infected with Lyme disease. And this is a study that was done in collaboration with Drs. John Alcott and Mark Solosky at Johns Hopkins. They have a very nice prospective cohort called SLICE, Study of Lyme Immunology and Clinical Events, in which patients are enrolled at the time of their diagnosis of Lyme disease when they present with acute fever and an EM rash, and they get followed for up to 2 years with serial laboratory testing, clinical evaluations, neuropsychiatric testing, etc. And we decided to focus on 3 key time points. So at the time of the first visit where a patient presents with fever and an EM rash, about 1 month later after a patient has had doxycycline treatment for presumable Lyme disease, and then at 6 months when some of the neurocognitive manifestations of Lyme disease can present. So we have 29 patients in this initial study, which has been published in NBio, along with 13 matched controls. And this study we're looking, and in fact in subsequent studies as well, we're looking specifically at peripheral blood mononuclear cells, or PBMCs, found in whole blood. And I want to acknowledge Dr. Jerome Bouquet in my laboratory who did most of this work. In our published paper, the interesting thing that we noticed, what you see here, this is actually a heat map, a collective heat map of patients presented with Lyme disease at visit 1, meaning they have acute fever and rash, and at visit 2, 3 weeks later following completion of treatment. What's actually quite striking is the fact that even at visit 2, when a patient presumably has the fever and the rash have disappeared, they're no longer symptomatic, majority of patients, and yet you can see here that there clearly is a strong inflammatory signature as shown by kind of the orange shading, the prominent orange shading associated with many of the inflammatory and infectious pathways associated with Lyme disease. And this was kind of a first hint, perhaps, that led to our hypothesis that perhaps we can develop a host-based test for Lyme disease. And especially because it appears to be persistent for at least several weeks. So the general idea is potentially, can we develop a diagnostic test in which patients present with fever and a rash, and it may have been several weeks since their exposure, and yet we can perhaps still be able to diagnose them with Lyme disease. And this is important because it really fits in what I call the window period, basically the 1 month period after the tick bite and the infection, in which we really lack adequate diagnostics. Now, after the window period, serology actually is pretty sensitive with respect to Lyme disease, at least IgG serology, but we really lack, and there are other problems with chronic manifestations of Lyme disease, but I really want to focus here on the window period. We really went into this wanting to develop a test that could diagnose infection during the window period of infection. So what we did was we took the data that we published and we basically analyzed them to develop a 172 human gene biomarker panel that appears to discriminate acute Lyme disease patients well from controls. So what you see here, these are 2 clusters, and these clusters are kind of generated kind of in an unbiased fashion, meaning I'm just taking the data and I'm allowing them to cluster together based on kind of similar properties across these genes. And if you basically generate a 172 gene panel, you can see here that it clusters into two groups, one group associated with Lyme disease and another group of matched controls. Interestingly enough, if you actually take several more clusters, so we looked at more data, such as data from patients with documented influenza, from patients that had other bacteremias in the hospital, some patients who had Mycobacterium tuberculosis infection. You can see here that they do appear to cluster as well, shown in this PCA plot, which is a principal component analysis plot. They cluster separately from the Lyme disease patients. So what we want to do next is we wanted to actually test this. Can we potentially optimize the panel? So we have 172 genes. I'll actually go into how they were generated from the data. But this is the general idea. We wanted to look at a large or several different patient cohorts. We wanted to look, for instance, patients with acute Lyme disease. We wanted to look at patients who are healthy matched controls, age, sex, and geography matched controls. We wanted to look at a cohort of, as well, patients with a different disease, tuberculosis, patients with acute influenza, patients with other bacteremias in the hospital. And we wanted to compare the host signatures corresponding to these patients against those with Lyme disease to try to optimize a panel that could be used for diagnosis. So I'm not going to go too much into this data. I can just tell you that it involves several different iterations where we had to basically generate, divide the patients into, for instance, both test sets, validation sets, as well as developmental sets. And what we did here was we eventually arrived at a way to select a panel that could be tested. And sort of implementing sort of what Dr. Dudley had talked about the use of machine learning, we actually adopted a machine learning-based approach to finalize the final deep gene panel. So we narrowed it down from 172 genes down to 86 genes that appeared to be pretty good. And then we took all of our samples, which total about 190 samples, 60 from acute Lyme patients, 36 of which were patients who were Lyme seropositive, 130 controls, both positive and negative controls, including matched healthy individuals, patients with flu, bacteremia, and tuberculosis. And then we basically divided them into a 50-50 into a training set and a validation set. And essentially we let the algorithm run, and the idea was to reduce the gene panel to the minimal subset of genes that could be discriminate most effectively Lyme disease from other diseases and controls. And we actually tested 10 different machine learning methods. So for those of you who are familiar with these approaches, there are many different ways that you can actually apply machine learning. There are many different algorithms that could be applied. And currently it's still theoretical as to which is really the optimal one. So we decided to just test all 10 that we had access to. It turned out that the generalized linear models, or the GLM models, turned out to show the best performance with respect to accuracy and kappa statistics. And in short, we ended up generating a 20-gene classifier. And what you can see there is, you can see in the misclassification error on the graph on the left, you can see that there's basically a trough, or kind of the lowest point, which is the least amount of error, was associated with about 20 genes. What you see on the upper right is actually a receiver operator characteristic, or a ROC curve, which then resulted in a final 20-gene panel that had about 94% sensitivity, 93% specificity, and about 91% accuracy for diagnosing Lyme disease patients. So what we're going to take as a next step is, the goal is actually to take this 20-gene classifier, and we're about to develop essentially an assay, a clinical assay that could be validated in the clinical laboratory. Okay. I also want to highlight that this approach is actually not specific for Lyme disease. So this is another example of where we actually did the same sort of human transcriptome profiling approach for diagnosing Ebola infection. In this case, we took, in collaboration with a group in the Democratic Republic of the Congo, we took samples of Ebola virus infected patients, whole blood, Lassa virus infected patients, and malaria compared to controls. And similarly, we could identify a 25-gene biomarker panel. This is sort of summarizing a lot of data. You see there that the 5 Ebola virus infected patients, they cluster in their own group. Notably, the patient with corresponding to the asterisk, that was a patient who was actually PCR negative for Ebola virus. And actually turned out, subsequently, he turned out to have Ebola virus infection. He was PCR negative probably because it was in the first 2 days, the eclipse period, before the virus is detectable in the blood. And this gives you an idea of like what we could do next, is that in addition to just identifying genes, what we can also do in addition to identify gene panel, we can actually identify specific genes that may be related. And so this might be a novel way of detecting kind of targets. So for instance, the final gene panel does contain some annexin genes, which have previously been described to be in association with Lyme disease. So this is a novel way of not only developing diagnostic panels, but potentially looking for new targets or new biomarkers of Lyme disease. Okay. I want to move into kind of the second kind of major omics area that we're studying with respect to Lyme disease. So we're really interested in doing precision diagnosis of acute infectious diseases using metagenomics. And the general idea is by applying metagenomic sequencing, we can actually directly make clinical impact for infectious diseases by providing essentially an organism ID with a turnaround time of hours to days. And it's really important that this be done in the setting of a clinical laboratory. So this is a test that has been, it's considered a laboratory developed test, or an LDT, meaning that it can be used for clinical diagnosis and management of patients. And what we want to do is target acute infectious diseases in hospitalized patients, including meningitis, pneumonia, and fever and sepsis, shown there. Part of the motivation is that those percentages are roughly the percentages of hospitalized patients who do not get a diagnosis despite extensive testing. And failure to obtain a diagnosis leads to delayed and inappropriate therapy, mortality, and excess healthcare costs. So it's our thought that what we can do is we can adopt what we call a metagenomic next-generation sequencing approach. The general idea is that you want to basically cast a wide fishnet. So instead of looking for a single target using a fishing pole, which is done by testing such as PCR testing, we want to send off a single test that in one swoop can detect kind of this full spectrum of organisms that cause infectious diseases. In principle, because all of these organisms are based on DNA, there should be no reason why a single sequencing-based test could diagnose all of these infections by direct detection in blood or other clinical samples. And this was highlighted in a case that we published a few years ago in the New England Journal of Medicine. This is Josh Osborne. He's a 14-year-old boy from Wisconsin. Went on a trip to Puerto Rico, came back and started to develop some headache and fever. Eventually was diagnosed with having an unknown meningoencephalitis, meningitis, and encephalitis. Ultimately underwent 3 hospitalizations over 4 months, spent 44 days in the intensive care unit, underwent more than 100 inconclusive diagnostic tests. He needed to get a brain biopsy as well because they still didn't know what was wrong, which was unrevealing. And he eventually developed intractable seizures. He needed to be placed in induced coma. It was an intensive care unit. And we received a sample on a Friday, and we were actually able to get an answer in 2 days, by Sunday. And it turns out, and this was him shortly after his brain biopsy. So we basically used a rapid bioinformatics pipeline called SERPI, which stands for Sequence-Based Ultra-Rapid Pathogen Identification. The general idea of SERPI is that you want to take millions of sequencing reads, be able to analyze and interpret that data for pathogens within minutes to hours. And we developed a clinical version of it, and this is what it looks like now. This is actually directly what you kind of see in the graphics visualization interface. What you see on the left-hand column is actually Josh's spinal fluid sample. And it turned out that he had been diagnosed with a disease called leptospirosis, caused by the bacterium Leptospira santarasi. What was quite interesting was that we subsequently sent his spinal fluid to the Centers for Disease Control, and it turned out that the CDC testing was negative for Leptospira. It was both antibody testing negative as well as PCR test negative. And you might ask, well, why was it negative? Well, it turns out that the CDC had not actually validated this species. This is a very rare species of Leptospira. It's only found in 3 areas of the world, including Puerto Rico. And so, as a result of this case report, the CDC has now updated their Leptospira. So I'm happy to say that they can actually detect this organism. And the treatment for this actually is penicillin. Interesting enough. He received both intrathecal and intravenous penicillin immediately. He was actually out of the ICU in 2 weeks and back home in good condition in 4 weeks. And he's doing really well now. He actually just came by to visit a few months ago. So we've actually leveraged this into a test that now has a 48 to 72 hour turnaround time. And this is really specifically a test for diagnosing meningitis encephalitis from spinal fluid. We have a series of clinical lab sequencing and then computational analysis. And now the results are reported automatically in the electronic medical record. This is actually a case of a patient who was infected by a virus called MX or MW polyomavirus, which you've probably never heard of, but it's there and we can actually report the result. So what we wanted to do is really explore the clinical utility of this test, and the idea is to take a clinically validated test and use a consortium of 7 hospitals in California nationwide, be able to enroll and consent patients for this test, and directly compare the performance of this test relative to all other conventional microbiological testing. And importantly, what we've done here is we also added the kind of a new feature, the clinical microbial sequencing board. This is a board that's modeled after a tumor board in cancer. And the idea is what we do is we recruit bioinformaticists, laboratory medicine physicians, infectious disease physicians, neurologists to kind of meet together, and we meet virtually over WebEx. But the idea is that we then discuss specifically the sequencing results and in light of the patients, and we provide kind of interpretation to the clinicians who order the test. What we were able to do over this 1-year study is diagnose neurological infection in about 20% of cases, but importantly, more than 1/2 of those were not identified at all by conventional testing. And it had pretty good performance with respect, and this is head-to-head against all other clinical microbiological testing. Now you may comment that as the diagnosis is low, but one of the criteria for enrollment for this study is that patients had to have no diagnosis at the time of enrollment. So typically these patients had been admitted, had been in the hospital for several days, and had had no diagnosis despite extensive testing, initial testing. This is gives you an example of kind of patient enrollment. So we ended up enrolling about 200 patients across 7 different sites. This is an example of a case report. I'm not going to go over too much more about the study. We're actually writing that for publication. There's a couple who both enrolled in the study. They went on a honeymoon to Maui in March of 2017. Upon returning to the US, both developed fever, a transient rash, headache, and neck stiffness. And the workup showed CSF eosinophilia. So they had eosinophils in the spinal fluid. Upon questioning, they said that they were hiking in the jungle and they were picking up and eating raw fruits. Any idea what this is? There was a state Hawaii advisory over this. Any ideas? Has anyone been traveling recently to Hawaii? Vacation? You probably heard of this. No? I think I heard it, yeah. So this is actually the rat lungworm, or Enterovirus cantonensis. It's a parasitic infection caused by, essentially, the life cycle involves snails, and so the idea is that you contaminated food and water, and you can actually get infected by this parasite. Interesting enough, this was, the patients were, the couple, they were enrolled in the study, and this was like a very weird result. We saw Enterovirus cantonensis. I actually thought it was cross-contamination of one sample to another sample. It turns out there were 2 different people. They were both infected with Enterovirus. Okay, I want to move into kind of an area which is directly related to what we want to do for Lyme disease and other tick-borne infections. So we want to actually implement nanopore sequencing. So what is nanopore sequencing? This is a pocket-sized sequencer where you can essentially do DNA sequencing directly from your laptop. So it's powered by the USB port in your laptop. We can potentially do DNA sequencing, RNA sequencing, and even protein sequencing. And the idea is that as these molecules basically they go through kind of these nanopores, changes in electrical conductance result in actual sequence data. And the other thing that makes this technology different than prior technologies is that you can do what we call real-time sequencing analysis. And I'll actually show you an example of it. So the idea is with traditional technologies like Illumina sequencing, you have to generate all of your data up front. And the turnaround time for generating data is typically about 18 to 24 hours. The problem with that is with infectious diseases, your patient might be dead by the time you actually get a diagnosis. So our idea here is we want to basically be able to run patient samples and generate data in real time that potentially could be actionable. And shown here, this is kind of a differential diagnosis of acute febrile illness. You can see that's very, very broad. And this is why we need to use something like metagenomic sequencing, because we don't want to develop primers and probes for every single one of these organisms. One is that it's going to be too expensive, it's going to be too impractical. There are challenges for actually developing specific targets. Instead, we want to apply a single sequencing test that can potentially diagnose all of this. And what I show there in red are actually the diseases that are tick-associated. So you see there are actually several of them which potentially could benefit from having a test like this. Some of the advantages in Nanopore sequencing is that you can do real-time sequence analysis. There's expansion potential for doing multiple samples at a time. There's long read capability, which we don't really use. We can also directly sequence RNA, so we've actually tested where some samples where we directly just sequence the viral RNA from patients with influenza, for instance. It's pocket size, and we've actually deployed this Nanopore sequencer in Brazil, in Mexico, and in the Democratic Republic of the Congo for doing outbreak surveillance, and potentially it has very fast turnaround times. There are some disadvantages. One is the current cost of sequencing, which I expect will go down as the technology becomes more widely adopted. The error rates are still fairly high, which tends not to be as big of a problem for us with respect to identification, and the quality of the flow cells can still be variable. But this gives you an idea of kind of the potential. This is a paper that we published in 2015 where we took a sample from a patient with chikungunya virus infection and a patient with Ebola virus infection. And you can see here, after starting the sequencing, we can actually identify these infections within 10 minutes. And it really illustrates the potential of this. Importantly, I'm not specifically using primers or probes to chikungunya virus or Ebola virus. I'm simply sequencing all of the data present, all of the DNA and RNA present in a clinical sample, such as a blood sample here. We've also been able to sort of leverage this by adding primers. Now, I may be contradicting, or it may sound like I'm contradicting myself because I say you don't need primers. Well, what's interesting is that you could actually take a metagenomic test. You can actually add targeted primers that are short primers. And in a way, you can actually improve sensitivity for detecting high priority targets, while at the same time not sacrificing the ability to detect things metagenomically. So this is a case where, for instance, I can take Zika virus primers in this case and be able to improve my sensitivity for detecting Zika virus, but I don't decrease the sensitivity for detecting HIV, which is not specifically targeted for. So what we've done is we've managed to harness these general approaches into a single test that has a turnaround time of about 3 hours, but that in principle can detect any organism. And we actually developed a new version of SERPI that can actually analyze the data in real time, so it can analyze about 1 million reads in 5 minutes. So the goal is actually, can we implement this in a point of care setting or in a clinical laboratory diagnostic setting? We've also managed to kind of develop them on these portable platforms. So it no longer runs on a big bulky instrument. You can actually run them on these miniature instruments. Ultimately, we want to develop a microfluidics device, such as the Voltrax, which is what you see there. That currently is not working well. It's sort of like a beta prototype. But ultimately, what I want is a push-button solution where you just put a sample down, push a button, and you'll get an answer within 3 hours. So this gives you an idea of kind of the real-time sequence analysis. This is actually an actual clinical sample. This sample has HIV and Zika virus. But you see here, after starting sequencing, you can actually detect HIV within 3 minutes. And Zika virus takes a little longer because it's lower titer. I think you might have to wait 13 minutes to detect Zika virus. This gives you an example of, so this is basically the 2 laptop setup where we basically are analyzing directly samples in Mexico for using Nanopore analysis. So we were doing dengue and Zika virus sequencing and detection here. This is a very busy slide, but all I want to highlight is that we've tested this now on many different body fluids, many different specimen types. You can see here we can detect strep pneumo from BAL fluid, staphylococcus from an abscess, Haemophilus influenza bacteremia, Plasmodium falciparum or malaria from blood, dengue virus from serum, Zika virus, influenza B from a respiratory secretion. So it's a general approach that appears to apply across a wide variety of different clinical samples and a wide variety of different pathogen types. So what we want to do here is that we've actually harnessed this into developing a panel that we call Tick-Seq or Blood-Seq. So the general idea is that we want to have high priority targets like anaplasma et cetera, and Babesia. We want to be able to harness that into a sequencing-based panel, incorporate the host-based biomarkers for Lyme disease as well, and be able to clinically validate that. So I just want to end by acknowledging the people in my lab on the left who did all of the work. Collaborators on the right, I'd like to specifically acknowledge the Stephen and Alexandra Cohen Foundation, as well as a new grant from the Department of Defense for developing an assay like this. It's sponsored by the Tickborne Disease Research Program. Thank you, and I'd be happy to take questions.